Thank you very much, Dr. Sonnenfeld and Dr. Greben, and also Martina for putting a terrific program for this meeting, and most importantly for the loyal audience who stays all the way till the end, which I greatly appreciate. So I have nothing to disclose, and I will tell you a little bit about our work about normal and leukemic hematopoiesis in the bone marrow in a dynamic microenvironment, and will also provide our point of view. So what you see here is a simplified model of how stem cells are regulated in the bone marrow. And you see here in purple the blood-forming stem cell, and you see in green the bone-forming stem cells. And they need this partnership because the blood-forming stem cells in a low metabolic state of reactive oxygen species low, in a quiescent mode of don't move, don't divide. This is how it is protected in the niche, and this is the reason why hematopoietic stem cells are chemotherapy resistant. So they need the partnership with the bone marrow stroma stem cells in order to be chemo and radiotherapy protected. However, we need billions of blood cells on a daily basis, and a wide variety of pro-inflammatory signaling induce secretion of the major chemokine CXCL12, which you see here as yellow spheres. So on the left-hand side, when CXCL12 is expressed on the surface of the bone marrow stroma cells, it induces adhesion interactions which maintain the stem cells in a quiescent, non-motile mode. However, pro-inflammatory cytokines like thrombin or GCSF will induce secretion of CXCL12 from the bone marrow to the circulation, and now the blood-forming stem cells can upregulate the major CXCL12 receptor, which is called CXCR4. And CXCR4, two major functions are for the migration of the cells as well as for the cell cycle. So now the stem cells can proliferate, differentiate, and egress into the circulation. However, in this state, they are no longer protected from radio and chemotherapy insult. As I've already told you, you can see the hematopoietic stem cell via adhesion interactions protected from chemotherapy by adhesion interactions which are induced by membrane-bound CXCL12. And in order for the stroma cells to present membrane-bound CXCL12, they need cell-to-cell contact and communication via gap junctions, in particular connexin 43 and connexin 45. And on the right-hand side, you see when the stem cells are free from the stroma interactions, they can elevate their ROS levels, and ROS high stem cells are only short-term repopulating and myeloid biased. So in order to remain undifferentiated, the stem cells need to lower down their ROS levels and to exit cell cycle in order to remain quiescent. As I've already told you, the hematopoietic stem cells which are adhering to the bone marrow stroma cells via CXCL12 and CXCR4 interactions are chemotherapy resistant. And on the right-hand side, you can see in the mouse model, connexin 43 knockout hematopoietic stem cells, and the mice are vital and are fine, and this connexin 43 knockout is only in the hematopoietic system. However, what happens when we inflict these mice with chemotherapy? So on the left-hand side, we see that ROS accumulates in the hematopoietic stem cells, and they transfer this ROS to the bone marrow stroma cells. So we can see that there is a cooperation and energy sharing in the bone marrow, and the hematopoietic stem cells, in order to remain undifferentiated and in a quiescent non-motile mode, they need to get rid of their excess ROS. In sharp contrast, in the connexin 43 knockout mice, this doesn't happen. The hematopoietic stem cells have ROS accumulation, which induces their either differentiation, apoptosis, senescent, and the mice are no longer protected from chemotherapy insult, and they die from hematology failure. And it's important to know why this can take place only in the bone marrow. After all, ROS is only like a gas can be released from cells. Why is this need for cell-to-cell contact? And one of the hypotheses that we're currently investigating is maybe we're talking about energy sharing in the form of mitochondria transfer. So it's not pure ROS transfer, but rather it's mitochondria transfer which maintains the stem cells undifferentiated, and I will get back to this point later on. As I already told you, the bone marrow stroma cells need cell-to-cell contact, and this way they maintain the stem cells in a quiescent, undifferentiated form. And we wanted to know what will interfere with cell-to-cell contact of the bone marrow stroma cells. And of course, osteoclasts are the first cells that come to mind. And you can see here that when the osteoclast digs the LUCANA, there is no more cell-to-cell contact between the bone marrow stroma cells. Membrane-bound CXCL12 cannot be presented, and then the stem cells will be free to proliferate, to differentiate, and to egress into the circulation. You see the hematopoietic stem cells in yellow. So we think that bone turnover and osteoclast-osteoblast interactions are needed not only for bone integrity, but we think they are needed also for blood production and for host immunity. And two elegant talks during this meeting, one by Toshio Suda and the other by Hans Snook, has revealed that hematopoietic stem cells have very low levels of calcium. And elevation in their calcium levels actually leads to their differentiation, and they lose their stem cell properties. And one of the reasons is the enzymes calpains, which are calcium dependent. They can degrade TET1 and TET2 and reprogram metabolically the hematopoietic stem cell differentiation. So we think that osteoclast-osteoblast interactions regulate bone turnover and host immunity. As you know, during GCSF-induced mobilization, there are elevated, intensified levels of osteoclast-osteoblast interactions. And these exciting preliminary results raise a big question: is what about leukemia-initiating cells, and what about malignancy? Are the malignant cells also sensitive to calcium levels, and will they respond as well? In particular, since we know that in leukemias there is some differentiation arrest, which is one of the problems. It's also important to know that the host stroma cells, which are important for clinical stem cell transplantation, while they can resist chemotherapy and total body radiation, which are the conventional preconditioning regimens for host patients in order to make them accessible for donor stem cell transplantation, these cells, while they are chemo-resistant and radio-resistant, they are sensitive to this treatment. And in this elegant study from the group in Switzerland, it was shown that bone marrow stroma cells are severely and permanently damaged by the preconditioning irradiation required for efficient stem cell transplantation. And what they did here, and this is with experimental mice, they co-transplanted with the donor hematopoietic stem and progenitor cells. They also transplanted intra-bone bone marrow stroma cells, and you can see in green the donor cells which are genetically marked with green fluorescence. And as a result, this doubled the levels of stem cell niches and stem cells that could engraft and repopulate, and as a result, it led to much faster neutrophil, platelet, and red blood cell production. So to summarize this part, we see that there is energy sharing in the bone marrow on a one-at-a-time basis. Either you produce new blood cells and new host immune cells, or you produced new bone cells. And you can see here on the left-hand side, anti-inflammatory cytokine like FGF2, KIT ligand, and CXCL12 itself will increase connexin 43 gap junctions and will help maintain the stem cells in a quiescent, non-motile mode, chemotherapy resistance. In sharp contrast, on the right-hand side, you can see that pro-inflammatory cytokines like the mobilizing agent GCSF or thrombin, they will disconnect connexin 43 gap junction interactions, and now the stem cells are free from the stroma microenvironment. They will have increased ROS levels, proliferate, differentiate, and egress to the circulation. However, in this situation, they are not protected from radio and chemotherapy and other DNA damaging insults. And it's important to know that the immune cells also participate in chemotherapy resistance. In these two studies, it was found that T regulatory cells, CD8 T cells, they can reside also in the bone marrow. And as you can see, their levels are increased following 5-FU. And on the right-hand side, you can see that via secretion of adenosine, they can lower reactive oxygen-less levels, the accumulation in the hematopoietic stem cells, and contribute to their chemotherapy resistance. The next topic that I would like to discuss is the topic of the blood-bone marrow barrier. We all know about the blood-brain barrier, the blood-retina barrier. There's also a blood-testis barrier. However, importantly, there's a blood-bone marrow barrier. And we know that mature red blood cells and mature platelets are found only in the circulation and not in the bone marrow. However, if you irradiate a patient for clinical transplantation or a mouse for experimental transplantation, you will transiently destroy this barrier, and red cells will penetrate into the marrow until the barrier is restored. And you can see on the left-hand side the barrier of endothelial cells, which can be either less permeable or, on the right-hand side, more permeable. And again, anti-inflammatory cytokines like FGF2 and downstream KIT ligand, they reduce the permeability, allowing for proliferation of bone-forming cells that you see here in green, and blood-forming stem and progenitor cells that you see here in blue. In sharp contrast, the pro-inflammatory cytokines will increase permeability of the barrier, and this will induce increased differentiation and egress to the circulation. In the mouse, we can reveal the architecture of the barrier, and we can look at the less permeable blood vessels that you see here labeled fluorescently in green, or the more permeable blood vessels. And if you look at the red dots on the top left, which stands for the levels of reactive oxygen species, you can clearly see that adjacent to the less permeable blood vessels, the ROS levels are lower, while adjacent to the more permeable sinusoids, the ROS levels are higher. And we think that this ROS comes from the circulation, from all the mature red blood cells and platelets, which can secrete S1P, thrombin, and other pro-inflammatory cytokines. So as a result, we established a model of the architecture of the barrier. You see here in blue on the top and the bottom, the more permeable blood vessels, the sinusoids, and they receive waves of metabolic signaling, reactive oxygen species coming from the circulation, encouraging the bone marrow to produce new mature red blood cells and host immune cells on demand. In contrast, you see that adjacent to the less permeable capillaries, arterioles, and arteries, they are coated by a second layer of stroma cells, and there the stem cells are in a ROS-low mode, both for bone and for blood production, and they are protected and shielded from all the high levels of mature red blood cells and platelets in the circulation, and we think this is one of the major reasons for a need for such a barrier. Interestingly, megakaryocytes are also mature cells which can regulate hematopoietic stem cells. They secrete a wide variety of factors, including TGF-beta, which maintain stem cell quiescence and a non-motile undifferentiated mode. And also interestingly, megakaryocytes were shown to inhibit osteoclasts. So in a way, next to the stem cells, there is a guard of a megakaryocyte protecting the niche from degradation and from turnover. And the next topic that I would like to discuss with you is the coagulation system. Although, as I've just told you, there is no coagulation in the bone marrow. There are no mature red blood cells, and there are no platelets in the bone marrow. And ancient Greek medicine, and it's called phlebotomy, mild bleeding. When a patient didn't feel well, he simply stretched his hand, mildly bled, and the patients claimed they felt better, like you do a blood donation now to a blood bank. And we have found that mild blood loss triggers osteoclast activation, and as I've already told you, osteoclast-osteoblast interactions can free the stem cells from their niche and allow stem cell mobilization. So there is some substance to this treatment because it replenishes the blood with new and mature cells. So we wanted to know if the coagulation system plays a role in hematopoietic stem and progenitor cells, as well as in the bone marrow microenvironment. So in mice it was found, and we know that hematopoietic stem cells are endothelial derived, and you see here in blue, in the bone marrow of mice, the most primitive long-term repopulating hematopoietic stem cells. These are the cells that when we transplant have the best long-term repopulation potential. They express an endothelial receptor which is called EPCR, endothelial protein C receptor. And it has been used as a marker in order to isolate stem cells and look at their repopulation potential. And indeed, we have repeated these results. However, we know that there are very low levels of stem cells also in the circulation for a very short time in order to avoid the high ROS insult. And you see them in green, and we notice that these stem cells do not express EPCR, and we wanted to know why. So there are two major coagulation cascades. One is the anticoagulant axis, where activated protein C can bind to EPCR, and together they cleave a peptide of the PAR1 receptor, and the cleaved peptide induces anticoagulant and anti-inflammatory signaling. And there are some research which is aiming at using these cascades also for the treatment of sepsis. In sharp contrast, on the right-hand side, there is a procoagulant and pro-inflammatory axis, where thrombin cleaves a different peptide of PAR1, and this cleaved peptide induces coagulation and pro-inflammation. So we wanted to know if these anti- and procoagulation cascades play a role in regulation of hematopoietic stem cells and their bone marrow microenvironment. We are also interested in downstream signaling of nitric oxide generation or nitric oxide inhibition, and nitric oxide is the smallest signaling molecule known. So you can see here in green, there is certain phosphorylation which induces the generation of nitric oxide, while in red, there's another signaling site which induces inhibition of nitric oxide signaling. So we know already that the hematopoietic stem cells are quiescent in a non-motile mode, protected from chemotherapy, with low levels of calcium, and we wanted to know their metabolic nitric oxide levels. So what you see here is the image stream which combines microscopy with flow cytometry. You see here in purple the EPCR positive stem cells, and you see in green nitric oxide, and of course you can appreciate that the most primitive stem cells have very low levels of nitric oxide as opposed to the more committed progenitor cells which have higher levels of nitric oxide. And as a result, APC, EPCR, PAR1 signaling, which reduces the levels of nitric oxide, also inhibits CDC42 GTP activity, induces CDC42 GTP polarity, and more importantly, downstream induces VLA4, which is a major adhesion molecule, expression, affinity, and adhesion. And you can see here the hematopoietic stem cell adhered to the bone-forming stem cells, and in addition to 6CL12, 6CR4 interactions, you can see that APC, EPCR, PAR1 signaling increases these adhesion interactions. So we wanted to know if EPCR signaling is at all important for hematopoietic stem cells, and you can see here in C on the right-hand side, We have treated mice with 5-FU chemotherapy and used either neutralizing or non-neutralizing EPCR antibodies. And you see that when we treated with non-neutralizing EPCR antibodies, all the mice lived. And when we treat with neutralizing EPCR antibodies, all the mice died, suggesting that APC, EPCR, PAR1 signaling are also essential, and that the long-term repopulating hematopoietic stem cells have an additional seat belt which ties them and links them to the bone marrow stroma cells, giving there more protection from chemotherapy insult. And what happened? And we wanted to know where is APC expressed in the bone marrow, and as you can see, protein C, which is the non-active ligand, is activated by another coagulation-related receptor, which is called thrombomodulin. And after thrombomodulin converts protein C to activated protein C can interact with EPCR and PAR1 signaling. And you can see that there are some blood vessels in the mouse bone marrow, in particularly the less permeable capillaries, which functionally express thrombomodulin. And perhaps interestingly and equally important, the hematopoietic stem cells themselves, the EPCR positive long-term repopulating hematopoietic stem cells, also functionally express thrombomodulin so that they can generate their own APC. Now, as I've already told you, APC, EPCR, PAR1 signaling inhibits nitric oxide generation. What about thrombin? So if we treat mice with thrombin, already within a few minutes we see increased levels of nitric oxide, including in the EPCR positive stem cells. And as a result, they activate a receptor, an enzyme which is called ADAM17, and ADAM17 is also expressed by hematopoietic stem cells. ADAM17 or TACE is needed for generation of TNF, and also has other important roles. It has an inhibitory motif that prevents it from being activated in the bone marrow. However, when the levels of nitric oxide increase in response to thrombin treatment, it induces EPCR shedding, it induces VLA4 shedding, c-KIT shedding, and other markers which are needed for maintaining the stem cells in a quiescent non-motile mode, allowing the stem cells to proliferate, to differentiate, and to be recruited into the circulation as part of host defense and new blood production. To summarize this part of the talk, we see here the two phases of the EPCR positive long-term repopulating hematopoietic stem cells. Chemotherapy resistant when they are maintained in the bone marrow in a quiescent non-motile mode with low levels of nitric oxide and high levels of VLA4 adhesion, which are providing with chemotherapy and radiotherapy resistance. And in sharp contrast, pro-inflammatory cytokines like thrombin, they increase the levels of nitric oxide, they induce EPCR shedding, and now the stem cells have higher levels of CXCR4 and of PAR1. They can proliferate and they can differentiate and egress to the circulation. So this is all shown in mice. As we know, in the immune system, the fetal liver hematopoietic stem cells have much higher repopulation potential compared to adult bone marrow. And this is a slide I received from John Dick, who did microarrays on human fetal liver compared to adult bone marrow. And you can see here the gene for EPCR, which is called PROCR. And you can see that the fetal liver stem cells have much, much higher levels of EPCR compared to adult bone marrow. Even the committed progenitors have higher levels. And Irv Weizman has told me the same is true also in the mouse fetal liver. So we think that with regards to EPCR and nitric oxide, everything that we have found in the mouse is also relevant for human stem cells. Many years ago, in the previous millennium, when I did my postdoc with John Dick, we developed functional preclinical models for normal and leukemic human stem cells. And we did it based on conventional stem stem cell transplantation using immune-deficient mice as recipients. You can see the mice are sublethally irradiated, and the human stem and progenitor cells are injected into the circulation, and then we can monitor for their population potential. And one of the reasons that we can have repopulation and functional assays in this cross-species model is because human and murine 6CL12 are cross-reactive. And indeed, we first found normal human stem cells, which we could identify and characterize, which give high levels to multilineage repopulation in immune-deficient mice. And later on, we found human leukemia-initiating cells, and I will elaborate on both. The group of Guy Sauvigeau in Montreal has shown that they can expand cord blood cells in vitro, and this leads to upregulation of EPCR, and all the long-term repopulation potential in immune-deficient mice is EPCR dependent. And the group of Haas in Germany has compared CD34 cells from mobilized peripheral blood compared to steady-state bone marrow, and they found upregulation of PAR1 on the mobilized cells, suggesting that PAR1 is needed for the mobilization process. And interestingly, there's a difference between fertile men and women due to the monthly period of women, which will impact on mobilization in response to G-CSF because of these PAR1 differences. And finally, CXCR4 is also a poor prognosis in human leukemias because its expression will give protection from radio and chemotherapy also to the malignant cells. And we have discovered that stem cells which have the same phenotype, leukemic stem cells, as normal human stem cells, CD34 positive, CD38 negative, they have poor prognosis. This is known from the clinic, both in pediatric and in adult patients. And you can see here the very high levels of EPCR in the human cell line KG1A, which was used by Kurt Sivin to identify CD34. And unfortunately, EPCR is a functional marker not only for human leukemias. Memory pad stem cells are also EPCR positive, and as a result, triple-negative breast cancer patients, which also have poor prognosis, have the same increased adhesion and chemotherapy resistance. Finally, when we look at AML in these immune-deficient mice, we see that the microenvironment is changed. There are increased levels of nitric oxide. The barrier is destroyed. The stromal microenvironment is destroyed. And inhibition of NO helps to protect and to allow normal stem cells to repopulate. And the same is true in other models of human AML, where also the ROS levels are elevated. And again, reducing ROS levels also helps in the chemotherapy resistance. So I would like to thank all our collaborators and thank all our lab members, and thank you for your attention.